Now it is my distinct pleasure to introduce our plenary speaker, Dr. Rosalind Wright, who holds the Horace W. Goldsmith Professorship in Children's Health Research and is Professor of Pediatrics at Mount Sinai Kravis Children's Hospital and Icahn School of Medicine at Mount Sinai, and Dean for Translational Biomedical Sciences. She received a Bachelor of Science in Human Genetics at the University of Michigan in Ann Arbor and obtained her medical degree from the University of Michigan Medical School, graduating cum laude. During medical school, she was selected to spend a year as a Howard Hughes Research Scholar at the National Institutes of Health in Bethesda, Maryland, further pursuing research in molecular biology. Following medical school, Dr. Wright completed an internship in internal medicine at the Beth Israel Hospital, Harvard Medical School. She then moved to Chicago, Illinois to complete her residency in internal medicine at Northwestern University, where she also served as chief medical resident. She then returned to Boston to complete her fellowship training in pulmonary and critical care medicine at Harvard Medical School. And if that's not enough, as part of this training, she obtained a master's in public health degree from the Harvard School of Public Health. Then she joined the clinical faculty at the Beth Israel Deaconess Medical Center and research faculty at the Channing Laboratory, Brigham and Women's Hospital, Harvard Medical School, and the Harvard School of Public Health, where she remained on faculty until she was recruited to Mount Sinai as the vice chair of clinical and translational research in the Department of Pediatrics. We are so honored to have Dr. Wright here. I am very excited for her talk entitled Elucidating Social and Chemical Environmental Determinants of Health Disparities, Promises of the Exposome. During her presentation, you're welcome to ask questions by way of live Q&A box that you will see on your screen and you can type, and she'll respond to at the end of the presentation during the Q&A portion. Dr. Wright, I'm passing the virtual microphone to you. Thank you.
Well, thank you very much for that introduction, and it's a pleasure to be here today to talk to you all and deliver this plenary address. Kick this off. It sounds like a very exciting meeting. So as Heather told you, I'll be talking about, she had given you the title. Oop. Somehow the slides are jumping ahead. I'll use my mouse. Can you see the slides?
Yes.
Okay, there we go. Okay, so as Heather mentioned, I'm actually an adult trained clinician, scientist, but why am I in the Department of Pediatrics? Because in my research life, I became increasingly interested in how far back I needed to go to put my patients and all on a healthier trajectory. And it turns out the roots of that start very early in life, regardless of what chronic disease you might be thinking of. And that we really need to take a life course framework and start at the very roots. So my work and some of the things I'm going to talk to you about today are looking at how early life exposures influence development in childhood and across the lifespan, and how we need to increasingly embrace the complexity of that, incorporating thinking around all the factors that might be operating there. As I said, multiple factors influence child development, even starting in utero, and this includes chemicals, pollutants, psychosocial stress. How you emotionally experience your environment is just as toxic, or perhaps even more so than some of the chemicals that we tend to think about. And then also nutrition, and we think about that as more of a resiliency factor, and I'll show some of that information. So historically, environmental health has really approached study designs by considering one chemical at a time, perhaps looking at lead and neurotoxicity, air pollution and respiratory toxicity, stressors, thinking about one type of stress at a time, racial discrimination, negative life events, something like this. What I hope that I'm going to convince you of today is that we really need to both measure multiple pollutants, chemical and non-chemical stressors in the same time to really understand the full scope of the impact of environment on chronic disease, as well as understanding the resiliency factors. And I'm going to do that in a particular disease paradigm. And what we're going to look at is prenatal programming by these environmental factors, multiple environmental factors on brain development in children. And you can look at a number of neurobehavioral, neurocognitive outcomes, of course, when you're thinking about that. But what I want to focus on today is some very interesting work we've been doing that exemplifies some of the framework that I'd like to advance in science going forward. And that's really looking at internalizing problems. Remarkably enough, people have looked at social stressors, programming things like depression, anxiety, which we know impact our society hugely. There are about 400 million people impacted by those disorders globally over the lifespan. And this, as well as many other developmental disorders, have their roots in this early period of development. And so we're going to measure things like temperament, negative affectivity, negative valence in infancy up to age 6 months, and then follow these kids and see if that translates to increased risk for internalizing symptoms, which get manifested clinically as kids age. And we're going to look at multiple exposures, chemicals, non-chemical stressors, metals is going to be a focus, as well as the stress, and we're also going to look at air pollution. So first, are children born to mothers who experience greater stress in pregnancy more likely to develop neurobehavioral cognitive problems? Again, when you're talking about internalizing problems, this has been the focus. It's more been on the social factors and psychosocial stress. This is a paper from our group. Rebecca Campbell was a former postdoc who's now on faculty at the University of Illinois, looked at prenatal stress in our pregnant women recruited out of the Mount Sinai Hospital across the New York the city, Manhattan boroughs, et cetera. And we looked at stress measured in a number of different ways. If you look at the left-hand side of this graph, there in the upper panels on the left, we looked at lifetime stress in mothers, predicted infant negative affectivity measured at 6 months. And you see a linear association, both with maternal lifetime cumulative stress, the next panel at the top on the right-hand side, current negative life events in the last 6 months around the pregnancy. And then the bottom panels look at psychological functioning in the moms, depression symptoms on the right, PTSD symptomatology, or I'm sorry, depression on the left, PTSD symptomatology on the right. And what you see is really a stark linear association between increasing number of lifetime stressors, current stressors, as well as more worse psychological functioning predicts negative affectivity in the kids. This is very consistent with what a broad literature shows us. Well, let's step back for a minute because we're talking about stress. Well, how does psychological stress get under the skin to influence health? When you experience psychological stressors in your environment externally, you weigh those against your own ability to cope with those. We bring different coping strategies to bear when we're faced with environmental challenges. And that activates key physiologic systems in our body. Most of us think of the stress response system as the HPA axis, the hypothalamic pituitary adrenal axis, which is shown on the left-hand side of this schematic, and cortisol production as the biological output of that influences some of the health effects of chronic stress. But if you look at the right-hand side, it's more integrated than that. The hypothalamic pituitary adrenal axis talks back and forth to the autonomic nervous system, and there's an optimal balance that the two of these want to strike to maintain health. But these also are integrated and talk back and forth with the immune system functioning and maintenance of oxidant-antioxidant balance. And this is the framework I tend to think about when I'm trying to elucidate some of the underlying mechanisms that might be operating for stress. But if you look at this schematic, which is kind of showing us that even if mom has disrupted physiologic functioning because of external stressors, that can translate to fetal health, which we're going to be talking about here today a lot, by disrupting the environmental milieu. Cortisol production, hypercortisolism is not good for infant brain development in particular. As well as other organ systems. And that's really through signaling through the placenta. And immune disruption, enhanced inflammation, enhanced oxidative stress, all of these things disrupt that maternal fetal signaling through the placenta, as well as other mechanisms that can play a role in brain development in starting in fetal life? Well, if you're studying the environment that we usually think about, the chemical environment, air pollutant, toxins from traffic-related pollution, for example, or other combustible sources, toxins in our environment like heavy metals, lead, arsenic, cadmium. We know those have toxic effects as well. And in utero smoking, which is shown in the cartoon up there in the upper left-hand corner, there's huge literature to show that moms who smoke, it has adverse effects on child development and growth, including the brain. They all push and pull on these same systems. Now, they don't overlap completely, but they affect similar disruption of these key regulatory systems, as does psychological stress. You can start to see where there would be cumulative effects or synergistic effects or interactive effects of those two. So we really need to think about them at the same time. And then down in the lower left-hand corner, I point to the fact that we're also going to mention briefly some resiliency factors like maternal diet to offset these toxins. So for example, increased antioxidants, offsetting, because we know oxidative stress is a key mechanism here. So that just gives you a high level framework of why we want to consider chemical and non-chemical stressors together. And this is the framing that we went into when we designed our cohort studies that I've now followed over a couple of decades, where we've measured. We start in pregnancy, we recruit women just out of their routine prenatal clinics. We don't go into any specialty clinics where we're looking at mothers who are particularly struggling with mental health issues or things like this. These are routine people presenting for routine prenatal care. And we measure during pregnancy and then following the moms and children's after birth. In the kids, we also measure these same factors: psychological stress exposure, ambient air pollution exposure, traffic-related pollution, mostly in the urban settings that we're doing these studies. We go into the homes and measure home allergens, nutritional intake for both moms and kids, and tobacco smoke exposure are the key factors, but we also biobank samples and we can look at toxins like metals and other organic chemical exposures, et cetera. And we look at a number of phenotypes, including neural development. Again, is going to be our focus today. And well, why do we measure both social and chemical stressors? We think a priori that they do push and pull on the same systems and that there might be multiplicative or synergistic interactive effects, but what's the evidence? So here's one of the studies that we've recruited out of the Mount Sinai Hospital, as well as Boston, the Beth Israel Deaconess Medical Center in Boston. As Heather mentioned, I had been in Boston before I came to New York City, so I do a lot of my work at the dual sites. And these are just putting people on the map, whether you're in New York City on the left, Boston on the right. They're coded by racial ethnic mix. You can see we have a very diverse population because we recruited out of communities where we could get that diversity. And you can see we have Black, Hispanic versus white Hispanic and white non-Hispanics, and that they live and cluster in different types of communities and neighborhoods. That's interesting to think about, and I'll show you why that may influence who's more likely to have greater level exposure to some of the toxins that we're talking about here. So now we have them mapped. We take their address, we can geocode them, convert that address to the location on a map like I just showed in the previous study. And then we can link that to various measures of the environmental factors that we might want to think about, whether it be air pollution, which is pictured at the top, or if you can look at the box in the lower right-hand corner. We also do other mapping other exposures like access to green space, crime or violence exposure in the communities, police record data, that kind of thing. We can also do access to healthy foods in a community, depending on what question you're going to ask. So this is a very powerful tool to use in our studies to really understand how the distribution of these environmental factors might be different, and maybe how they play a role in why are we seeing greater burden of certain health outcomes, including neuropsychological dysfunction in particularly lower income, primarily ethnic minority populations. That's the idea of designing the study. Here's one of the exposures that we've used a lot in our studies, is looking at daily exposure to air pollution, traffic-related pollution, because these are urban samples. That's a factor that they are likely to be exposed to. And we look at fine particulate matter, which is what the PM 2.5 stands for here. And this is Dr. Alan Just, who's one of our junior faculty here at the Icahn School of Medicine at Mount Sinai, who does this kind of modeling, coordinating satellite data from NASA, satellites with ground monitors that are there for the EPA. And you can get very high resolution exposure for each individual pregnant woman. And then we can use that in some of our epidemiologic studies. And here's a paper that came out just recently where we looked at prenatal exposure to this traffic-related fine particulate matter, and we linked it to this measure of infant temperament that we were at age 6 months. Infant Behavior Questionnaire revised version is what we're using. This was developed by Mary Rothbart, and it's the most widely caregiver reported measure of infant temperament. And what we were focusing on is, as I said earlier, was this measure of negative affectivity that you can get off one of the factors from this questionnaire. And that can be broken down into a number of features, which are shown here. Kids who demonstrate more sadness types of behaviors, distress to limitations, fearful behaviors or inhibition, don't like novel situations, that kind of thing. You can pick that up already in a very young infant, and then how quickly they can recover from distress. And what you see is that we don't see a main effect for the negative affectivity related to prenatal PM 2.5, but when you look at it within these features, we see a very strong effect on fear behaviors, and that's in both boys and girls, interestingly, when we looked at the air pollution exposure. And remarkably, again, this was the first time people had looked at chemical exposure affecting temperament in young kids. But now, why would we want to do it? Because we know negative affectivity is a particular temperamental trait that is robust and tracks with later risk of internalizing behavioral disorders, has some effects on externalizing behaviors as well, like conduct disorder, ADHD, but what we're going to focus on is likelihood of going on to develop anxiety and depressive types of disorders. And so negative affectivity is certainly linked to that. And fear, interestingly, is a construct within that that's related more to anxiety disorders. So we were interested to see, well, are there other chemical environmental influences that might affect risk for temperament disorders in our infants? So we said, well, we can measure a panel of exposure to metals, heavy metals that we already know from the literature are related to brain development and some adverse neurodevelopmental outcomes. And they're pictured here. So we looked at arsenic, barium, cadmium, chromium, cesium, lead, and antimony. This was measured in maternal urine for these analyses. And what I'm showing you there is the distribution of the exposure to these chemicals within our pregnant women recruited out of both Boston and these New York City maternal prenatal clinics, and comparing that to the geometric mean value for pregnant women enrolled in the National Health and Nutrition Examination Survey, a US representative sample just for reference. So you can see that our moms who live in lower income, largely lower income urban environments are more highly exposed relative to the NHANES data for most of these. And then we took a look, because we're interested in understanding if differential exposure to the environment impacts health disparities in these communities. We looked at the distribution of these metals in our pregnant women based on a number of individual level factors, which are pictured in this schematic. And basically what I want you to focus on, there's a lot of data there. This is published, so you certainly are welcome to go and get more insight from the paper that's listed there. But what we saw, and not unexpectedly, because that's what a lot of the literature shows, in our pregnant moms, if you had less than a high school versus college or high school education versus college, you were more likely to be more highly exposed to a number of these metals. Also Black and Black Hispanic versus white non-Hispanic, and the Hispanic non-Black versus the white non-Hispanic were much more likely to be exposed to multiple heavy metals. And you can see that very clearly here. Relative to our white non-Hispanic moms, both Hispanic non-Black and Black Hispanic had much higher levels of a number of these metals. But we also looked at this in relationship not only to individual level factors, but community level factors. So where you live also matters. And I won't go through all of this again, because these are published, but basically when you look and you're going to communities with higher crime index, a marker of the lack of resources invested in those communities for a number of reasons, the percent population with high school or less education, et cetera, they were much more likely to be exposed, in addition to those relationships with the individual level, SES and other markers we looked at before. So that's interesting. That just gives you a sense of, I think, we really do need to understand the distribution of, differential distribution of environmental factors that we think are driving some of these health inequities based on neighborhood, but also individual level socioeconomic and demographic factors. And then we took a look at, in the same women, at this exposure to the metal. And here we didn't look at one metal at a time. We used advanced machine learning, regression modeling, weighted quantile sum regression modeling that can account for all 7 of these metals, even though they're correlated in exposure, et cetera, and look at a mixture of them as the index of the exposure and predict then negative affectivity on this infant behavior questionnaire, as we did in the previous work with air pollution. And what do we see again? And really, I want you to focus on the left-hand graphic, which shows you that fear, again, was this particular feature of negative affectivity that was driving the effect, the relationship with increased adverse temperament outcomes in the kids. And it was really different for boys and girls. It was particularly in the girls. And this is interesting because now we're starting to see fear increased in girls in relationship to this metal mixture. And we know that girls are much more likely as well to go on to develop things like anxiety disorders. So it'll be interesting to follow them and see if that's a pattern that carries through as these kids grow up. Let's remind ourselves of the mechanisms that we're thinking about, because I want to show you some work about looking at how race, ethnicity, and your exposure to stress across the life course really plays a role in how the physiologic functioning of some of these key regulatory systems are operating in these pregnant women as they head into that pregnancy. So a lot of it's related to all the stress over their life might bring into that pregnancy. And so this is a paper from some time ago in our earlier cohort that we recruited out of Boston, similarly to what I described for the PRISM study, across a number of communities, very ethnically diverse. And we took saliva from moms during pregnancy. So we asked them to give us saliva samples immediately in the morning, within 45 minutes prior to brushing their teeth, and then prior to lunch, prior to dinner, and right prior to bedtime. And then we looked at their diurnal cortisol rhythm, which is a measure of their stress responsivity over the course of the day. And we related that to their history of exposure to a number of types of stressors, which are shown in that upper right-hand corner there. Exposure to interpersonal violence. So this was history of exposure to interpersonal violence as a child abuse, in dating violence, domestic violence in more adult relationships prior to the pregnancy, and even including the pregnancy, as well as community level violence. So not only what you're experiencing personally, but how about in the communities that you live in? And then ongoing discrimination, and then other negative life events at the time of pregnancy. And what we showed in the bar graph in the lower right-hand corner, if you look at our African American and Hispanics, even though they're similarly living in communities with lower SES, less education, et cetera, our African-American women tended to be more highly exposed to these, more than one of these cumulative stressors. And that is what rocked the HPA axis, which is circled in the red on the left-hand side. It was really amongst our African-American women who had been more highly exposed, where you saw a very atypical flattened rhythm over the course of the day. And this is something that's been related to depression and multiple adverse outcomes in the patient themselves, the participant themselves. But also, if mom has this abnormal rhythm, hypercortisolism produced over pregnancy, it's going to affect the kid. Another way to measure cortisol in mothers is with hair samples. And this is a more cumulative measure of cortisol production over the time period of hair growth. Hair grows about 1 centimeter a month. So what we do in our pregnant moms when they give birth, we collect hair as shown in the pictures at the bottom of this slide, cutting right at the scalp nearest section, tied off with a, and then you can count back by centimeters and assay for cortisol based on what you're interested in. We're interested in trimester exposure, so we do a 3 centimeter scalp closest segment of hair to represent the 3rd trimester, for example, which we're going to talk about in some of the analysis that we show. And here we published a paper again some time ago showing that it's very different in white, Hispanics, or African-American mothers in our cohorts, where how they're entering pregnancy, what their levels of hair cortisol looks like across trimesters. And you can see African Americans have more hypercortisolism relative to the Hispanics, relative to whites. And on the right, a lot of that is driven by differential exposure to early life abuse, child abuse, emotional abuse, physical abuse, sexual abuse, as you see there. And that's driving a lot of the stress effects in our population, as well as disrupted physiology that might interact with these other chemicals that they're also have a higher burden of exposure with. So let's try to put this together and think about looking at interactions, interesting interactions or synergistic effects. Take a more complex picture of what the environmental factors might be driving the differential outcomes, and how we can better identify the true, more precisely, who is really at risk. When you look at things like lifetime cumulative trauma exposure and predicting negative affectivity in our infants at 6 months, we really don't see a main effect, an independent effect of that that's significant. But if we say, well, how about a mom who has lifetime cumulative trauma elevated and shows disruption of these key physiologic systems, in this case, hair cortisol levels, and then relate that to temperament. And what we showed in this paper is that it's the interaction that's interesting. And as you see increase, a rug plot of increasing cortisol exposure over the in our moms in a 3rd trimester hair sample, and as you know, the exposure to lifetime trauma has increasing impact on that negative infant negative affectivity as you get to those higher levels of cortisol. So it says you have to understand these more complex interactions. There are sex differences there too. We won't go into that, but those are talked about in the paper. Now we're going to sort of try to look at metals and cortisol activity. Do those interact to have interesting effects on this measure of neural behavioral outcomes in the kids? And for this, we used, again, the machine learning WQS repeated holdout method to look at this complex interaction between this, adjusted for the factors that you see there. And we did find significant 3-way interactions, even in a sample of less than 300 maternal child pairs, which says to me this is a very strong interaction when you can pick it up in a sample of that size. And we found that there were some significant interactions for negative affectivity, the overarching composite measure, as well as fear, sadness, and falling reactivity features of that. And it really was the mixture of these multiple metals was really showing an effect, a significant effect, particularly in girls exposed to high levels of cortisol, as you can see in the bottom right-hand graph. But it was fear that was driving it, which is, you can really see here in the lower right-hand graph, where you see a very stark difference between girls who were born to mothers who had low levels of cortisol production over the course of pregnancy, more typical cortisol productions. Even if they were exposed to the metal mixture, you didn't see much of an effect, but you sure see a huge effect in the green for those born to mothers with high cortisol. The graphic just shows you, in the mixture model, you can also go use that WQS mixture model to tell you weights of what are the metals that are driving it. And that'll be interesting when we want to inform, go a little deeper on mechanisms as well as interventions that we might want to think about. Another interaction that we looked at was this prenatal metal mixture and stress mixture interactions on anxiety problems, now measured in these same kids at age 3 years. So now we're looking, as they get a little bit older, we can more definitively measure things like the construct of anxiety that we're interested in seeing if that's an emerging profile over the course of early childhood. So again, you can identify it early and do something about it so you don't have chronic problems with this anxiety over time. Here we had to work with our biostatisticians and our computational scientists to develop new methods that could account for this prenatal metal mixtures, the multiple metals considered at a time, interacting with our stress measures. Here we're going to show in the lifetime stress, cumulative stress measured in panel A, and current life event measures with the crisis measured in panel B, which are current during the last 6 months. What have you experienced during this pregnancy? And what you can see, and we use this new kernel machine approach that could really put this data all together and tell us what profiles look like. And again, we're predicting anxiety problems in these kids at age 3 years using the child behavior checklist in this case. And what we see is, if you have lower level or intermediate level exposure to the two stressors, you do see an effect as you increase your exposure to this metal mixture. You see those graphs, those lines go increased risk for anxiety problems, blue and green lines. Look at the red line. If you're someone who's exposed to high level of cumulative lifetime stress and or current contemporaneous stress in the last 6 months during pregnancy, it's really the stress that's driving the effect. And you don't see on top of that, at least the way we're measuring it, additional effects of the metals. So to me, that suggests if you ignore the social factors, you won't get much bang for your buck by intervening only around chemical toxins like these metal exposures. So I think that's an important takeaway from that. And certainly there's more work we need to do, and there's lots of interesting things we can ask, and we are for that, as well as other groups. Now, the other thing I wanted to pivot again and think about, well, even on a more complexity, okay, we know higher stress is toxic, metal mixtures, air pollutants are toxic, even starting in utero on brain development. But what about if we throw in and think about resiliency factors as well? Not just look at risks, but we really life and health and maintaining this optimal balance is really a balance between your risk factors and your resiliency factors at the same time. And certainly during these key developmental periods, it's going to be important. I mentioned at the beginning, particularly antioxidant intakes might be protective of things like PM 2.5, as well as metals, because they operate one key underlying central mechanism might be oxidative stress. And this is just a paper that suggests that that indeed is true in our population. So now we're going to pursue an analysis where we look at cumulative exposure to these multiple stress measures, lifetime stress, current contemporaneous stress measured in the last 6 months using the crisis. And then we're also going to look at maternal psychological functioning in utero. So her score on a depression scale, the Edinburgh Depression Scale, or PTSD checklist. And we're going to use stress mixture modeling, similar to how we've used it and applied it. And more people in environmental health have applied it when you're thinking about chemical mixtures like the metals that we've been talking about. But we actually asked, could we also use this to look at stress mixtures? And indeed, it turns out it's a nice way to do that and to more comprehensively categorize or characterize stress in these mothers as a mixture. And that couple of papers there will go into more detail in that. We again use the air pollution exposure here, PM 2.5 that we talked about before. And now we're going to fold in antioxidant intakes in the moms during pregnancy, which we measured with a food frequency questionnaire was validated using 24-hour recalls in our moms, a couple of papers describing that validation there. And then we actually wanted to look at antioxidant micronutrient intakes in particular, and all of those listed here: beta carotene, vitamins A, C, E, zinc, magnesium, and selenium. And then again, create an antioxidant mixture, account for all of those, right? Because that's the reality. Moms aren't just exposed to one or the other or have high intakes of one or the other, but we really want to know a more complete picture of antioxidant intakes. And then we asked our computational scientists to help us do bigger, use bigger and bigger data. And what we looked at, what we were really interested in is at the bottom there. How can we examine mixture by mixture interactions when we have 3 mixtures in the model? And here we're looking at repeated wheeze. We haven't done it yet for the temperament and some of those outcomes. I'd hope to have that today, but I'm going to use that as sort of a proof of concept model here. And they developed this new, again, another machine learning advanced regression modeling, Bayesian kernel machine regression, or BKMR. And here's what you get from that. You can look at across these 3 factors, their mixtures of antioxidants, the stress mixture, and the PM 2.5. And this is on the sample overall, estimating repeated wheeze. And you can see lower intake of antioxidant on that index is related to higher increased risk for wheezing. Increased stress is associated with increased risk for wheezing. PM 2.5 doesn't seem like much is there. It's really flat in that right-hand panel. But if you look at within African Americans, they're the ones, again, that are particularly at risk for all 3 of these. If you have lower antioxidant intake, higher risk for wheezing in children. Higher stress, higher risk. And then there's a trend toward higher PM 2.5 exposure and higher risk. This is just a visual, how you can visualize. There's, these are complex 3-way interactions of mixture by mixture by mixture. So that I don't mean to have you look at those and be able to interpret those right away, but you can visualize it. And here's what we eventually showed. In our overall sample, it was really an interaction between the antioxidants and stress that was driving the effect. But if you look within African Americans, it was all combinations of this. Decreased antioxidant, increased stress, as well as decreased antioxidant, increased PM 2.5, and the stress and the PM 2.5 were interacting. So those all were interacting to enhance the development of wheeze. Not so much in our Hispanic population, as you can see there. So again, you're trying to get a little closer to defining who is at particular risk. And why is that important to do it, and do it as early in development as possible? Because if we can actually identify these risk factors in a preclinical state before they manifest, what you and I are seeing for anxiety disorder, sleep problems, whatever comes from that, you have more opportunity and likelihood of changing that trajectory. Right, their kids tend to continue to be very plastic, and it doesn't sort of get embedded as a trait until later. So as early as we can find them, it's important because it's easier to build strong children than to repair broken men. I have this quote hanging in front of my desk. It reminds me why we do the work that we do. So in order to ask and answer these more complex questions of the environmental influences cumulatively that are affecting health, we really need to change our perspective. And here is the pictorial view of what I've been describing. We're really evolving towards an exposomic framework. Exposomics and I'll talk about it in some of the other slides going forward. We're really trying to say, let's as comprehensively as we can be able to measure, assess, and analyze the effects of multiple external exposures that are driving these health outcomes, because we know they interact in complex ways, and we need to understand that to understand the full scope and impact of these. And we need to figure that we have to think about both social, non-chemical, as well as chemical stressors, some of which I've given examples of today. And that's embedded in this life course framework. We have to think about it at these critical windows of development and how that might change in our thinking of what the mechanisms might be and what the intervention and prevention strategies might be. So if we're really going to disentangle health disparities, we really have got to embrace this notion of how complex the external environment is and the driving factors is shown here in this cartoon. And there's lots of new ways emerging to measure that, some of which I've shown you here, such as the air pollution modeling, harnessing things like the satellite data from NASA, et cetera. And how do we scale it up to really be able to measure effectively, efficiently, cost effectively the totality of exposures from preconception to death, or whatever time point that we're looking at? You really don't have to measure all of it to really make headway here. We're already making headway with just some of the examples I've shown you today, and we're not measuring all of it, but we're measuring it more comprehensively. Measure as much as you can, if not the totality, and we need to integrate that into the way that we're doing our science, and we need to evolve our computational science right along with it. And we're really there. We're really much more farther along than many people might think. It's really a paradigm shift to a new systematic understanding of the cause of disease. This, as genomics was, there was the decade of genomic science. This is the decade of exposomic research. I think the emerging technologies are there. The computational science is there. The other component that I didn't talk about today, but this makes me think of it, looking at this schematic, is we need to think about gene environment interactions in the context of what we're talking about as well. And that's important. Well, how do we do that? We're going to need to expand measurement of the external development, harness emerging digital technologies. I showed you some of the geocoding approaches that we're using to be able to identify it in patients. We also have to be bigger and bigger in how we characterize the internal environment, the internal response to that. I showed you some that are much more targeted measures, like cortisol that we measured in a couple of different ways. We need to do that on an omic scale. We have to understand how all hormone systems may be disrupted, for example, not just cortisol, related to a lot of these different environmental factors. And that's what's pictured here. Is it's not only thinking on a grander scale for measuring the external environment, but exposomics is also thinking on a greater scale. And how do we design science and biobank, et cetera, accordingly to be able to really do that with the biological response to these environmental exposures as well. That will give us early signals, early profiles, early fingerprints of who's at risk. And we have started to do that. I'm not going to show these data today, but we've incorporated this exposomic type of framework. Some of the omics types of internal biological response functioning that we're looking at in relationship to these external environmental factors are using proteomics, immune phenotyping, epigenomics, metabolomics. These are some of the tools that are available to us at our Institute for Exposomics and more broadly to investigators, increasingly so, as we go forward and try to address these complexities of environmental programming of chronic disease. Again, this reminds me, and all of us should take a breath and not get overwhelmed and say, you don't need to measure it all to make significant progress. And this cartoon of Moses on the Mount thinking, well, how about we start with these 10 and see how it goes? So you have to start somewhere, and you can make significant progress even if we don't sort of realize the complete conceptualization of describing all environmental factors that are in, health relevant. Another thing that I'd like to make you aware of, there are resources that have been stood up knowing that this is where our science is going in environmental health. And the National Institutes of Environmental Health Sciences have invested very heavily in what's now known as the Human Health Exposure Analysis Resource. And the goal of this is to provide infrastructure for adding or expanding exposure analysis to advance understanding of the impact of environmental exposures on human health throughout the life course. So if you are a study who has been focusing perhaps on only the social environmental factors, or it's a genetic study, but you have samples banked, you could apply to this HERE network to, at no cost to you, if your application is accepted, to have some of these environmental factors measured, whether that's on some of the omics scales, metabolomics, et cetera, or you want to do more targeted, I want to measure this panel of metals, et cetera, to understand if it's impacting some of the health outcomes I'm interested in. Here's what it looks like. They funded this series of laboratory network across the US, and Mount Sinai has both the untargeted and targeted, but the others are there at University of Minnesota, Wadsworth, RTI, and Duke. And you can access it through this website, the HERE program. You're also welcome to link through to our Exposomic Institute. We also have the Data Coordinating Center for HERE at Mount Sinai. So we certainly can answer your questions and direct you to these resources more directly. Another resources that's been invested in by the National Institutes of Health is the ECHO program, the Environmental Influences on Child Health Outcomes. I'm sure most of this audience is familiar with this. This was applications that allowed a number of us to apply and come in and be all in on merging our data across 82 US cohorts. The ones that I talked to you about today are in there, for example, in our consortium. And what we want to do is harmonize and bring together up to 50,000 mother-child pairs, families, and biorepositories to be able to ask and answer some of these more complicated questions that we're looking at with a much larger sample size, which gives us power to really go after this whole exposomic framework in a meaningful way. Another thing that I'd like to bring to your attention, is educational tool that was developed out of the CTSA programs, the Clinical Translational Science Awards at the National Institutes of Health, funded through the National Center for Advancing Translational Science. And it was really a program developed, bringing together experts to develop educational webinars and tools to help people wrap their head around charting the life course, exposomics being a part of that. And that is what I contributed to this whole panel of experts that were brought together to do this, to say, well, how do we think about really scaling up exposomics in this context and taking advantage of the evolving exposure science as well as computation science. So that I'm going to stop there. Of course, there's a number of people, some of who have been mentioned during this presentation, but many more who have not, who have helped build this transdisciplinary science that I'm talking to you about today. We get bigger and bigger, and we have to bring in greater and greater expertise to do this together. It's been very exciting, and we've been fortunate to have support through a number of the funding agencies that I've talked to you about today, including the ECHO Consortium and the CHEER and HERE lab networks. So I will end there and take questions.
So we do have several questions, so I'll hold my own and ask the audience. The one interesting question asked about looking at twin studies, and I think that's really interesting. So I'd be curious to hear what you thought.
Yeah, we do not include twins in what I was talking to you about. That's actually an exclusion criteria. Multiple pregnancy is an exclusion criteria for the ones we have there, partly because it would be difficult to sort out some of the things we're looking at that I talked about. But there are interesting twin studies. that I know of, that I've actually been interested to read about, showing that if you have twins and you have male and female, in that you can show that boys and girls are very differentially sensitive to certain disruptions in physiology during pregnancy, like oxidative stress. Boys seem to be more vulnerable to oxidative stress in utero. The placenta is a dimorphic organ at the time that it starts to grow during pregnancy. And you can see very different signaling in that pregnancy related to boys and girls relative to these mixtures of environmental factors. So twin studies will help us inform that. We need more of them to ask and answer these specific questions, but there are a few out there that suggest that it will be interesting to pursue that. So that's a good thought and a good question.
Another question asked, maybe if you could elaborate a little bit more about epigenetics and how it plays into the exposome.
Yeah, so epigenetics, the increase we've seen in a number of these developmental disorders, including neuropsychological disorders, neurocognitive disorders, can't be genetic alone, right? Because we don't mutate our genes that fast. It's not a genetic mutation, but epigenetics is really a chemical change in the genome that results from exposures in the environment. That's been shown over and over in studies, whether it's air pollution or metals, et cetera. They can actually change the chemistry, methylate, for example, genes in certain areas where they now turn off and turn on differently during development. And that, as you can imagine, it can be a powerful mechanism to alter your ongoing development and trajectory. Some of those are advantageous, and they're trying to adapt to the environment that the fetus, for example, is developing in, but they can have long-term adverse consequences. They can result in, adverse clinical outcomes, unfortunately. And that's how we use epigenetics. We can show that the lifetime cumulative stress changes the epigenetic profile differently in our boys and girls. We're looking at things like microRNA expression from the placentas in these kids, that kind of thing. That's another epigenetic mark, although some people argue about whether that should be an epigenetic or what. But that's an important signal to think about. So that helps show you biologically how it gets embedded, right? You know this exposure, this is the profile of the epigenome. So it's a good way to look at this early risk. And then there are others. Mitochondriomics is another one that people are doing now that's quite interesting, for example.
Unfortunately, we're at time. There is one more question that perhaps if the anonymous person who asked this could chat it into the Attendify, or we can get it to Dr. Wright if you email it to myself through the Attendify. It's asking about why primarily internalizing behavior. I don't know if you want to really quickly mention why you think this is in the exposure, and then we're at time.
Right. Yeah. Why that? Because there's a gap in our understanding. As I tried to point out, I was surprised myself when I went to the literature and that people were not thinking about chemical factors programming internalizing behavior. We spent so much time thinking about lead and neurocognition. I started with IQ, but then it became more behavior, and then it looked at conduct disorder and attention problems, memory problems, those kinds of things. But it's very important to understand what are the roots of neuropsychiatric illness. Particularly think about the pandemic with the economic fallout and the social distancing. We're going to see an epidemic of anxiety and depression. It has its roots in early life, and it manifests by early childhood heading into adolescence. And that's what we're trying to do with this work. I use it as an exemplary, right, outcome. You could have used this to look, I could have talked to you today about asthma risk, lung growth, lung function growth and development, obesity, cardiometabolic. It was just meant to be exemplary, but it's also a huge gap. That's why.
Thank you very much. Thanks so much. You definitely did not disappoint. Really fantastic talk, taking really complex factors and how they relate and making it very understandable. So appreciate it so much. Thanks to our program chair, Dr. Jay Shrinair, and co-chair, Dr. Vivian Yap, the planning committee, AFMR leadership, chair of the Eastern region, Dr. Laura Castleman, and chair-elect, Dr. Andrea Hahn, and meeting manager, of course, Taylor Baumgartner, and to all of you for attending, and of course to Mead Johnson Nutrition for making this session possible.